Hello everyone, welcome to my channel EaLi's Teaching. Now I discuss about the blood cells and how blood cells are formed. Please don't skip any part of this video for clearance of the topic. Discussing about the blood cell. What are the types of blood cells? Here three types of blood cells are present. Number 1 is red blood cell or RBC. Another name is erythrocytes. Then number 2 is white blood cell or WBC or another name leukocytes. Then number 3 platelets or another name is thrombocytes. They play an important role in our body. So important discussions about blood cells, how these blood cells are formed. How these blood cells are formed. Blood cells formed by the process hematopoiesis. Here, three types of hematopoiesis present. One is erythropoiesis, second one is leukopoiesis, and third one is thrombopoiesis. By the process erythropoiesis, RBC formed, and second process leukopoiesis, by which WBC formed, and third process thrombopoiesis, by which platelets formed. Most important topic is RBC. So I discuss about RBC, all the related questions. First of all, RBC, it is a simpler structure than most human cells. RBC are extremely important to your health because RBC carry almost all the oxygen your body uses. And structure of the RBC is biconcave disc-like structure. Structure of the RBC is biconcave disc-like. Then I tell you about the erythropoiesis process, how the RBC formed or the erythrocyte formed. Now I tell you about the erythropoiesis process. Erythropoiesis means by which generation of RBC. So, how this process occurs? Stages of the erythropoiesis. First one is proerythroblast, from where early normoblast, then intermediate normoblast, then late normoblast, from where reticulocyte, and then mature erythrocyte. Showing picture about the process of erythropoiesis. Here, stem cell to the proerythroblast and proerythroblast to early normoblast, then early normoblast to intermediate normoblast, and from intermediate normoblast to late normoblast to reticulocyte, and after 2 days reticulocyte to mature RBC. First one is proerythroblast. It is the first cell derived from stem cell. It is very large in size with a diameter of about 20 micron. Its nucleus has 2 or more nuclei and a reticular network. Proerythroblast does not contain hemoglobin and the cytoplasm is basophilic in nature. Proerythroblast multiplies several times and finally forms the cell of next stage. The next stage is early normoblast. Early normoblast, this cell is smaller than the proerythroblast with a diameter of about 15 micron. In the nucleus, nucleoli disappear. Condensation of chromatin network becomes dense. Cytoplasm is basophilic in nature, hence the cell is called basophilic erythroblast. Early normoblast develops into the next stage called the intermediate normoblast. Cell is smaller than the early normoblast. Diameter 10 to 12 micron. Nucleus is still present, but the chromatin network shows further condensation. hemoglobin starts appearing, cytoplasm is already basophilic in nature, cell develops into the next stage, late normoblast. Late normoblast. Diameter of the late normoblast decreases and diameter is about 7 to 10 micron. Nucleus becomes very small with a very much condensed chromatin network. It is also known as ink spot nucleus. So, a important question is what is ink spot nucleus and which cell contain ink spot nucleus? Then answer is late normoblast cell. In the late normoblast cell, quantity of hemoglobin increases. Cytoplasm becomes almost acidophilic, so the cell called as orthochromic erythroblast. In the final stage of the late normoblast, just before it passes to the next stage, that is called as reticulocyte, and the nucleus disintegrates and disappears. The process by which nucleus disappears is called pycnosis process. It is also important. What is the pycnosis process? Pycnosis process by which nucleus disappears. So, in the late normoblast, two important questions: what is ink spot nucleus and what is pycnosis? Now I tell you about the reticulocyte. Reticulocyte, it is also called as immature RBC. It is slightly larger than the matured RBC, and the cytoplasm contains reticular network. Due to presence of reticular network, it is also called as reticulocyte, and then it convert to matured erythrocyte. In matured erythrocyte, reticular network disappears and the cell becomes matured RBC and attains biconcave shape. The cell decreases in size to 7.2 micron in diameter, and the matured RBC is with hemoglobin but without nucleus. It requires 7 days for the development and maturation of RBC from erythroblast. So, pro-erythroblast to matured RBC, 7 days. And it requires 5 days up to the stage of reticulocyte. So, pro-erythroblast to reticulocyte, it requires 5 days. And from the reticulocyte to matured RBC, it requires 2 days.